Hello and welcome to the February Research Information Volunteer Call on Cell Therapies. Today's presenters are Esther Latros, PhD, AVP of Research, and Jaime Giraldo, PhD, Associate Director of Research.
Thank you, Brian. We look forward to providing an update on cell therapy. This presentation follows two previous great updates by Frank Martin and Simi Ahmed that cover universal screening and disease-modifying therapies, our two other priorities under the CURES program. Upcoming RIVs will cover updates and priorities, including improving outcomes in people living with type 1 and supporting training of researchers and clinicians. As a reminder, T1D progresses through distinct stage, and our vision is to have cure therapies available to all affected by or at risk of type 1 diabetes. Starting at how can we identify individuals at high risk of developing type 1 using diagnostic tools like autoantibodies to beta cell antigens that can develop before any clinical symptoms of T1D. The team nicely discussed our focus on disease-modifying therapies that prevent, delay, stop, or reverse the type 1 diabetes disease process once the autoimmunity has developed. While disease-modifying therapies at stage 2 and 3 are directed at preserving beta cell function and restoring glycemic control, the focus of today's presentation is on replacing insulin-producing cells at a stage that no functional beta cell remains. So from islet transplantations, we know that replacing beta cell function in the established diabetes is an approach with a clinical proof of concept that demonstrates full glucose control and insulin independence can be achieved. Importantly, we refer to cures at different stages. And while we analyze gaps, risks, and opportunities differently at each stage, I want to remind and keep in mind that our research efforts in these three areas are highly connected. As you heard from Frank, what we do in screening will impact disease-modifying therapies, and what we do and what the advancements in disease-modifying therapies can actually advance our efforts in the established stage. For instance, in efforts in regeneration or immunotherapies, even though it's not reflected in the slide, definitely some advances in disease-modifying therapies can be applied to later stages. Finally, the other two areas that I want to emphasize that overlap across our CURES project and priorities includes, number one, investigating how the immune system recognizes beta cells. This is important in autoimmunity and also important in, for instance, preventing rejection of donor insulin producing cells. And the second area of our effort is how do we bring advances in technology in many other fields to type 1 that can be applied to diagnosis. You'll hear about gene editing, antibody technology to target some tissues or cells, and biomaterials. So with that, I'm excited to pass it on to Jaime.
Thank you, Esther, and hello, everyone. So I will be speaking about the cell therapy project today. What are we referring to when we speak about cell therapies? These are therapies that use live cells to treat or reverse disease. At Jdrf, the cell therapy program refers to delivery of externally derived beta cells or islets to restore insulin independence and blood glucose control. And so this is a big distinction between cell therapies and what we commonly refer to as beta cell therapies, which are part of the regeneration program, where these are therapies targeted towards beta cells to reduce their stress and induce their growth to restore the lost beta cell mass. And so I just wanted to take a moment to make that distinction and avoid any confusion. And so Jdrf is accelerating the development of cell therapies towards commercialization by supporting preclinical and clinical testing and development of these therapies, providing support to gain regulatory approval for these therapies, and finally access to these therapies. So where are we today? Today, we have a clinical proof of concept where islet transplantation has been shown to provide a cure, a functional cure for established type 1 diabetes. So this consists of the transplantation of cadaveric donor islets into people that undergo immunosuppression. Now, there are some challenges associated with transplantation. Number one, we have a very limited supply of these cells, so we can't treat everyone with type 1 diabetes. And number two, it requires chronic immunosuppression, which carries risks such as the risk for opportunistic infections and malignancies, which limit the application of this therapy to people that suffer from severe hypoglycemic events and hypoglycemia unawareness. So currently we have a few stem cell-based cell therapies in clinical testing, and so we have some promising prototypes there. But toward the future, we would like to develop cell-based therapy products using a renewable source of cells and with iterative advancements. And so over time, we'll see these products improve in terms of increasing the duration of insulin independence, reducing the need for immunosuppression, and reducing the need for encapsulation, which I will talk about in more detail later on. In terms of the cell therapy project, we have two main components of the strategy. One is to develop a reliable source of beta cells for implantation and also support their survival and function after implantation. And the second is to shield the beta cells from the immune destruction. In terms of developing an alternative source of cells for implantation, the idea here is to really have enough cells to treat everyone and make sure we have access to those cells. Now, they can be derived from stem cells, human stem cells, or from animal sources such as porcine islets, as you can see in the figure on the right-hand side. Now, each of these sources has advantages and disadvantages. In terms of the animal-derived sources, they are more abundant, but they can provoke a stronger immune response. There is a risk for transmission of animal diseases to humans, and we are not sure which specific stage of maturation these cells should be used at, whether it's, for example, neonatal pigs, recently born pigs, juvenile pigs, or adult pigs. In terms of the human stem cells, of course, these are human-derived, so they don't generate as strong as an immune response as those cells derived from other animals. However, they do cause concerns for safety, as they can develop teratomas, which are benign tumors, if they are not properly differentiated. And we still can't generate mature beta cells that secrete insulin just like adult cadaveric islets. And so we're also trying to find out what is the optimal stage of development where these cells need to be implanted to have optimal function. And so Jdrf is supporting the development of both of these sources of cells and derivation methods to develop these products. Now, another aspect that we have to think about is where do we implant these cells? Currently with islet transplants, these cells are infused into the circulation of the liver where they lodge in the blood vessels. And so this is not ideal. They're in direct contact with blood, which causes blood clots and increases the immune response. It can cause inflammation of the liver, which also induces an immune response. Now, the liver processes a lot of toxic chemicals, and so it's a toxic environment for the cells, and it makes it impossible to retrieve the cells in case there's a need for that. So as a result, we lose more than 50% of the cells transplanted in only the first few days after transplantation. And so we need to find a better site that provides nourishment and enables retrieval, if possible, of these cells. Now, having blood vessels is key. One, to nourish the cells, provide them with oxygen and all the nutrients that they need to survive, but also to enable them to sense the glucose and secrete insulin back into the bloodstream in response so they can regulate the blood glucose. And so Jdrf and our supported projects are exploring various different sites, including the skin, muscle, and the abdominal cavity. And so these have all different advantages and limitations in terms of access. The skin is superficial, it's easy to monitor and retrieve if needed, but also in terms of the immune reactivity and the amount of blood vessels present. And so we are currently exploring different sites to determine which one is the optimal one. In terms of how do we protect the cells, there are various approaches that we're pursuing. One is encapsulation. So encapsulation consists of using a physical barrier that blocks the immune attack while ensuring cell survival and function. And so you can see a diagram on the right, on the top on the right, which is a macroencapsulation device. You can see that immune cells are bouncing off on the outside because they're being blocked, whereas nutrients such as oxygen and glucose on the right-hand side penetrate through the device, and insulin and other hormones are secreted outside of the device. Now, with microencapsulation, it's one device to deliver a large amount of cell clusters. So this is desirable, it's very practical, it simplifies the surgical procedure, and it simplifies retrieval of the device if necessary. Now, there are limitations to the device in terms of the transport of oxygen, nutrients, and glucose and insulin due to a lack of blood flow within the device. And therefore it relies on diffusion, which is basically just molecules floating around, moving from high concentration to low concentration, which is very slow and only works for small distances. So this becomes a challenge with larger devices. And as a result, the field has moved towards microencapsulation, which is using smaller capsules, as you can see on the lower left, that each contain a or a few cell clusters. Now, this increases the surface area and decreases the volume, so it improves the transport to the cells of nutrients and other factors. But it does complicate retrieval since you have to implant a larger number of capsules. This has also been improved more recently to a method known as conformal coating, where we coat individual clusters with a very thin coating, as you can see in the figure in the center, which improves the transport of nutrients even further. And then finally, we have nano encapsulation, which is depositing ultra thin biomaterial coatings on the cluster surface. And so this also results in even further improvements in terms of the transport, but also is associated with challenges with retrieval. In all of these approaches, we do have to be careful about the fibrotic response. Anytime we implant any foreign material into the body recognizes it as foreign and creates this fibrotic capsule around the implant, which basically chokes off the cells. And so we have to make sure that we use highly biocompatible materials to avoid this. And these materials are typically very stable so that they don't degrade once they're implanted. Now, another strategy that we're pursuing to protect the cells from the immune system is immune modulation. Here, the idea is to alter the immune response to the implanted cells and or device, thus allowing the cells to thrive. And so here we use biomaterials for controlled delivery of drugs and or presentation of helpful immune signals at the implantation site to direct the activity and function of immune cells interacting with these materials. So as you can see in the figure on the left, you have an artificial antigen presenting cell. It's just a biomaterial. And you can see that there are these little blue molecules being secreted out of the cell interacting with the immune cell, which is the T cell on the bottom. And so that gives signals to the T cell in terms of how to behave. We can do this through these soluble signals that are released by the biomaterial, or through these other signals, as you can see here, that look like a Y, tethered to the biomaterial surface, which also interacts with immune cells and directs them in terms of what to do. And so the idea here is to use these strategies to suppress rejection of implanted cells by suppressing immune cell function, or we can actually use these to also re-educate the immune system to accept the implanted cells. So this is a very novel and exciting field and area that we're exploring as well. Finally, we're also looking at gene editing to protect the cells. And so the idea here is to use new and novel tools to enhance cell survival and function of implanted cells by editing their genes. So just to give a brief background, genes control cell function by controlling which proteins these cells produce. And so we can intervene by targeting genes and by way of the genes, the proteins involved, for example, in immune recognition, autoimmunity, and tolerance. And so as you can see in the figure on the right, on the left-hand side, you have on the top a cell, such as an implanted beta cell, that has these markers, these proteins on the surface that the immune cell, which is on the bottom, recognizes. And so we can use gene editing to knock off those markers in the transplanted cells, which is what you see on the right, where the immune cell does not recognize the implanted cells, but does recognize these other signals for acceptance. And so this is how we can use gene editing to camouflage the cells from the immune response. We can also target different genes and proteins that make the cells more robust and enhance their function so they survive in harsher environments or reduce the stress of the cells once they're implanted. Finally, we're also exploring an interesting technology, which is scaffolds. And so these kind of fall in between the immune protection and the enhancing survival of the cells after implantation. These scaffolds enable engineering of the local environment to keep the cells alive and serve as a multi-purpose platform. They're basically a porous matrix, as you can see in the figure on the right. It's like a sponge, which has these open pore structure. And so these are used to distribute and house the cells. We can't have the cells clumping together because they'll compete for nutrients and oxygen with each other. And so we want to make sure they're distributed throughout the entire implant. Now, the open structure allows for infiltration of blood vessels to provide nourishment for the cells and enable their function, detection of glucose and secretion of insulin. And we can also use these to deliver oxygen to support cell survival during the time that these blood vessel networks grow and develop inside this implant, or also to deliver drugs to promote that blood vessel growth. Additionally, we can use these scaffolds for immune protection in the sense that we can use it for local delivery of drugs or presentation of immune signals, as I just mentioned, with immune modulation, or they could also be used in combination with micro or nano encapsulation approaches or genetically modified cells. So what is JDRF's role in cell therapy? So JDRF supports research and development across the entire development pipeline, from early discovery, where we invest in novel and exciting areas that are high risk but have the potential for high reward, through translational studies where we advance promising technologies from testing in small animals towards large animals and clinical testing, and eventually reaching the clinic where we test these therapies in humans. Now, throughout this process, we also have our advocacy team, which we work closely with to provide advice on regulatory strategy to ensure that these trials are approved and we can actually enter the clinic for testing. Finally, we also work with both academia and industry and play a role in bridging the two with the goal of developing commercial products. So we support research that in academia that's focused on discovery and development of novel therapies and strategies. We catalyze partnerships between academic investigators and with promising technologies and industry partners capable of developing commercial products. They have the infrastructure, the means, and to develop these products for commercialization. And we also directly fund industry to develop these products. And at the same time, as I mentioned earlier, we work with our advocacy team to provide support for regulatory strategy to ensure that these products are approved. Another approach that JDRF has taken to accelerate the development of these therapies is the establishment of the beta cell replacement consortium. And so development of these therapies requires scientific and technical expertise from multiple areas. For example, engineers can design devices, but they may not know how to test them in animal models, or they may not know as much about the cells, the beta cells. And so JDRF created the Encapsulation Consortium in 2013 to facilitate development of these cell therapy products for T1D. There are more than 50 members in the consortium, and they all range in expertise from beta cell sources and stem cells to bioengineering, animal models, biomedical and translational science, transplant medicine and surgery, and immunology. And the consortium also includes key industry players with commercial capabilities. The consortium serves as a platform for the exchange of research data, reagents, and protocols, and has led to the establishment of several successful collaborations, the development of multiple encapsulation technologies, and significant advances in islet transplantation and movement of early stage research forward to translational development. Moreover, the JDRF has also pursued centers of excellence. And so the idea behind these centers of excellence is to supercharge efforts and drive accelerated development using multidisciplinary science. And so here you see a diagram with the 4 centers of excellence. The Michigan Center of Excellence you see at the bottom is focused on improving beta cell survival by improving their energetic and metabolic capacity. The second Center of Excellence, which is in the Northern California area, is focused on immune tolerance by modulating the immune system of the person receiving the cells and the immune response. The third Center of Excellence, which is the New England Center of Excellence, their main goals is focused on immune modulation, not from the side of the immune cells, but from the side of the beta cells. And how do we modify the beta cells to prevent that immune reaction? And finally, we have the Center of Excellence at the University of Wisconsin, which focuses on creating immune evasive beta cells as well. Now, in terms of progress, before I start speaking about this section, I'll preface it by saying that we will go over some of the projects in the portfolio and not the entire portfolio, as we don't have enough time. These are some highlights of very exciting recent developments, but we have several other exciting projects ongoing. For ViaCyte, I think this is one that we get asked about a lot. It's a very exciting project. They're very exciting projects, multiple going on at the same time. As you can see, they have 3 products in the development pipeline. The original product, which is PEC and CAP in the center, is an encapsulated cell therapy, is in clinical testing. And we have the PEC Direct, which is similar to the PEC and CAP product, where this pouch that carries the cells has larger holes and so allows the infiltration of blood vessels to nourish the cells and provide nutrients. And finally, the PEC QT, which uses a similar pouch, but uses genetically modified cells for immune protection. And that project is earlier in development. In terms of key progress, clinical studies have demonstrated that the cells mature and produce insulin and C-peptide after implantation. And these experiments provided key findings regarding the surgical procedure in terms of implanting these devices. Now, the PEC Direct trial data is very promising, and they're working on optimizing the approach. These trials are ongoing now. And ViaCyte entered a clinical phase agreement with Gore, which is a partnership they've established based on a novel membrane technology for this PEC and CAP product. And so clinical evaluation of this PEC and CAP product has resumed using this novel membrane, and preclinical and initial clinical data suggests that the novel membrane can mitigate this fibrotic response I was speaking of earlier. Vertex is another very interesting new development in terms of, it's a leading pharma company that acquired a T1D fund company, SEMA, which is pursuing beta cell therapy. They have 2 parallel programs. One is a cell implant with immunosuppression and no device, and they're hoping to enter clinical trials in early 2021. They have filed their IND very recently, as they announced at JP Morgan in this month. And also an encapsulated cell implant with no immunosuppression, and this product is still in preclinical development. Next, we have Cernova, which is a Canadian-based company, which we are funding as well, and is pursuing a scaffold-like approach. And the idea is to prime the skin prior to implantation to enhance the survival of the cells. And so we implant this scaffold-like device. It promotes the growth of blood vessels at the site. And then subsequently, we insert the cells, as you can see the figure on the right, in these little hollow chambers that have been prevascularized to support cell viability and function. Now, they demonstrated promising preliminary clinical trial results where no adverse events were observed. The device was safe and well tolerated. The device integrated well and demonstrated ingrowth vessel networks, and indications of clinical benefits such as reduction of daily insulin requirement and C-peptide in the blood was observed. More recently, we heard the announcement from Novo Nordisk and Procyon Technologies. They established a collaboration, and so this is very exciting. It's a big pharma company investing in a collaboration for the development of an encapsulated cell therapy. The approach here in this project is to provide oxygen to the macroencapsulation device to enhance the capacity that in terms of how many cells the device can carry and improve the cell survival. And so this pairs Procyon Technologies' oxygen-enabled encapsulation device and Novo Nordisk stem cell-derived insulin-secreting cells. This collaboration also came out of the consortium, actually, and so the focus is going to be on further optimization of the device and the cells to accelerate preclinical development and entry into the clinic. Jdrf played a critical role in supporting the early development of these devices in the technology and in catalyzing this partnership, as I mentioned, through the consortium. Next, we have this project by Andres Garcia and Habal Sherwan. And this is an academic collaboration developing a biomaterial-based immune modulation approach. And so the idea is to use biomaterials for local delivery of immune signals to suppress immune rejection. So Andres Garcia is a bioengineer at Georgia Tech. Habal Sherwan is an immunologist at the University of Missouri, formerly the University of Louisville. And they showed that by incorporating these biomaterials with the cells, after implanting these materials with the cells, they demonstrated prolonged survival of the cells and acceptance and long-term function when they combine this with a short-term immunosuppression, basically combining it with delivering immunosuppressive drugs for only the first 2 weeks after transplant. This partnership has been established with an industry partner, iTolerance, to accelerate clinical development of the technology. And it's a potential platform technology that could use additional signals, additional immune signals to modulate the immune system and enhance outcomes. Next, we have the Kisler Yi Project at Joslin Diabetes Center. And so this is an approach where they are using gene editing to avert autoimmune responses to implanted cells. And so the team has identified multiple genes that make mouse beta cells vulnerable to autoimmune attack. And so by blocking the function of the top candidate gene they identified, they showed that the cells are protected from autoimmune attack. Now they are looking at testing combinations of these genes, blocking combinations of these genes to enhance the effect and validate the approach using human cells. And so the idea here is to use gene editing to modify the beta cells prior to implantation to avert autoimmunity. And another avenue that we could pursue is also using drug-based therapies to block the activity of these genes. So in terms of key takeaways, what is the main focus of the cell therapy project? The two main priorities are to ensure that we have alternative sources of beta cells available for implantation, approaches of delivery to ensure that the cells survive, and approaches at immune protection to protect them from the immune response. Jdrf is supporting research across the entire pipeline to accelerate development and clinical translation of these therapies. And we are balancing mature and novel next-gen technologies in the portfolio. So supporting early research that's starting and later research closer to the clinic. Now, Jdrf also supports research in both academia and industry, and we catalyze the establishment of these new partnerships to accelerate development and commercialization of these technologies. Multiple companies are showing very promising early results in the clinic and gaining critical experience in doing so to move development of these therapies even further. And we have several novel alternative approaches to immune protection aside from encapsulation that are also yielding very promising results.